Good morning, everybody. And I would like to introduce Akash to all of you, who I'm sure many of you have met throughout the summer, but Akash comes to us from Seton Hall University. And it was a pleasure working with him this summer. He's going to talk to you about a protein, HMGB1, and its role, its potential role in pediatric dilated cardiomyopathy. Our lab is interested in studying differences between the family pediatric heart and the adult heart, and so Akash is going to show you some of those differences and what he learned through the summer. So thank you, Akash.
Thank you for the introduction, Dr. Miyamoto. My name is Akash, and today I'll be talking about HMGB1 mediated ectodomain shedding and via ADAM17 in pediatric dilated cardiomyopathy. So to begin, I'll be discussing what dilated cardiomyopathy is, which is in short for DCM. DCM essentially involves the enlargement of the left or even both of the ventricles. As we see in the picture above, we see a picture comparing a healthy heart compared to a heart with DCM. This enlargement of the ventricle causes a weakening of the ventricle, and as we know, the ventricle is, its speciality is to pump blood throughout the body, and thus the body is not as efficiently pumping blood in patients with DCM. So what is the significance of this disease? The primary significance is that this is the most common type of cardiomyopathy that is found in children, as about 40% of pediatric patients that are diagnosed with DCM require transplant within 2 years of diagnosis. So what are the causes of this disease? So these DCM can have a lot of genetic underpinnings, but oftentimes the cause is idiopathic, which means that we don't know the reason of what is causing this disease. So now I'll be going into myocardial fibrosis that occurs in DCM. Myocardial fibrosis essentially refers to the collagen buildup that is occurring in the heart muscle cells, which is essentially cardiomyocytes. The impact of this is that the heart becomes extremely stiff, which leads to diastolic dysfunction, which essentially refers to the heart's inability or its decreased ability to relax. And we see that diastolic dysfunction is a very powerful prognostic indicator for DCM. As DCM disease progresses, diastolic function decreases with it. This is in contrast to ejection fraction, which decreases at the onset of the disease, but there are very limited treatment options to diastolic dysfunction in contrast to ejection fraction, which is why I'm going to be researching on this. And now I'll be talking about the distinct features that are present in pediatric DCM in contrast to adult DCM. So one of the main characteristics that is different is there's very limited cardiomyocyte hypertrophy that is present in the pediatric populations in contrast to the adult population. The second main difference is that there's a very different pattern of myocardial fibrosis that occurs. In adult populations, we have interstitial fibrosis, which means that the collagen is woven in between the heart muscle cells or cardiomyocytes, while in pediatric population, the pattern is more endocardial, which means that the collagen is in the inner lining of the heart chambers itself. And also the third main thing is a lot of treatment options have been geared towards adults, as a lot of research has been done on adults with DCM, and thus, as we expect, children don't have as robust a response to these heart failure medications compared to the adults, highlighting the need to study this in even more detail. So now I'll be going over the protein of interest, which is known as high mobility groove box 1 protein, which is in short for HMGB1. HMGB1 is essentially in every resting cell, or most of every resting cell, and it's localized in the nucleus of the cell, and it regulates gene expression. But when the cell is under stress or necrosis, HMGB1 is passively released, and it becomes a damage-associated molecular pattern. So what is the relevance to this protein in pediatric DCM? So to understand this, we must have some context. So for some context, the University of Colorado extracted protein serum levels of patients that have non-failing hearts, patients that have recovered hearts, and patients that required a transplant and had DCM. And it was seen that looking at the HMGB1 levels of the serum, we see that HMGB1 levels were extremely increased in patients that required transplant compared to the patients that had non-failing hearts or even recovered hearts, highlighting that we need to study this in even more detail. So with this, I logically hypothesize that HMGB1 promotes profibrotic gene expression. So the experiment that I used to test this was that I first cultured primary cardiac fibroblasts that were isolated from neonatal rat hearts. Then I used treatment conditions, the first condition being the control, which was essentially untreated. And then I had the second condition be TGF-beta, stands for transforming growth factor beta, which is a well-known positive control. And then I treated cells with HMGB1 with varying concentrations to see its effects with 10, 50, 100, and 200 nanograms per microliter. These conditions were imposed for a total of 48 hours. And some of the assays that I used included qPCR, which was used to analyze gene expression of genes alpha-SMA, periostin, and collagen-3, which are known to be fibrosis-related genes. And then I also used Western blots to detect ADAM17 expression levels, which I'll be talking about more in detail in the coming slides. So now the results that I got for these three genes are shown in the graph. Although we see that TGF-beta, the positive control, didn't work as intended, and thus this data cannot be extrapolated, I suggest we look at the trends to further our understanding. So looking at the trends, we see from HMGB10 to 50, there's a great increase in gene expression. But then from 50 to 100 and 200, there's a great decrease. So why is there an increase, then a decrease in gene expression with higher concentrations of HMGB1? And this leads us to ADAM17 as a potential regulator of HMGB1. So ADAM17 is essentially a shedding enzyme that cuts the receptor proteins along the cell membrane. So then we thought that ADAM17 maybe is shedding RAGE and TLR4, which are the receptor proteins that HMGB1 binds to when HMGB1 concentration or levels elevate. And this contributes to the early fibrotic remodeling that is present in the pediatric DCM patients. So to further test this, we used a Western blot, and we did a similar treatment with HMGB1, but we had untreated, we didn't have TGF-beta, but we had HMGB10, 50, 100, 200, and 400. And we see a similar trend to the qPCR, where the ADAM17 levels are increasing from untreated 10 to 50, but then it's decreasing from 50, 100, 200, and 400. These results look much more promising, and thus we could see that when HMGB1 levels are below a certain threshold, we see that fibrosis gene activation is activated and it's increasing. While this is occurring, ADAM17 expression levels are increasing simultaneously with that. Once we reach this certain threshold, and past this threshold, ADAM17 essentially takes over, and it cuts these receptor proteins, and this leads to a decrease in HMGB1. This leads to a decrease in fibrosis gene activation because of increasing concentrations of HMGB1 past the threshold. So now with this, with the data that I've extrapolated, I would now like to go into the future research directions that could be made out of this data. So we see that in the qPCR, we see that HMGB from 10 to 50 is increasing, and then from 100 to 200 it's decreasing, but then we also see an increase in 400. So why is that occurring? We don't know, and thus that is something that we could research even further. And then the second thing we could do is we could investigate HMGB1 The other receptors involved in ectodomain shedding, including RAGE and TLR4, and we could do a Western blot on these and see the effects of it. And the lab is currently doing this as we speak. And the third and final thing that could be extrapolated or done is we could compare the neonatal ADAM17 expression levels to adult models and see how that differs and varies. So now in conclusion, I've shown how increasing concentrations of HMGB1 increase, then decrease fibrosis gene expression, and we see that ADAM17 is a key regulator, or it's promising to be a key regulator of this, and in turn, ADAM17 protein levels are increased then decreased with HMGB1, increasing concentrations of HMGB1, which is very consistent with the qPCR results that have been obtained. So now I would like to give a huge thank you to the CHRI lab. Especially I would like to thank my mentor, Dr. ADAM17, for being there since day one. It would not have been possible to do this without you. I would also like to thank Dr. Miyamoto for giving me a clinical perspective and showing me around the clinic and all that. And I would also like to thank the Children's Health Research Internship, especially Dr. Dahl, Dr. Foreman, and Claire for hosting this and allowing me the opportunity to present my research. Thank you. Any questions?
I have a very speculative question. Is it possible that the expression of ADAM17 is also related to electrical activity in the heart, simply because ADAM genes in the brain are actually associated with electrical activity?
I mean, that's interesting. I have not really looked into electrical activity in the heart and stuff. I've just looked at DCM, so I'm not fully aware of that. But I guess that is something that could be done in the future. So thank you for bringing that idea up.
Do you have any predictions about what would happen when you compare to adult ADAM17?
I would think that just because of, I think the ADAM17 levels should, stay the same, because I don't think ADAM17 is regulating the activities. Amanda could correct me if I'm wrong, but yeah.
So the idea is, the initial idea is compare adults.
The idea is compare adult and neonatal fibroblasts. So why? Because the fibrosis in pediatric population is different. So probably ADAM17 in adult population, the regulation via ADAM17 is lost, and we have increase of this kind of fibrosis, interstitial fibrosis. And in neonatal, I think it's suggesting that the regulation of fibrosis is working because we have high levels of ADAM17 that can regulate these receptors. Otherwise, HMGB1, that is a DAM molecule, RNA molecule, can drive, exacerbate fibrosis. So the hypothesis is that ADAM17 is regulated in pediatric population, and during the increase of inflammation in adults can lead to the loss of this important regulation via.
Hi. Are you aware of the clinically relevant dosage of HMGB1 that is present in clinical studies?
I don't believe there is a clinically dosage of HMGB1 yet. I'm not aware of that. I don't think HMGB1 is prescribed drug or something that could be used. But it could be something in the future that could be prescribed. So.
And my second question is, what was the concentration of TGF-beta that you used in the study?
TGF-beta, we did 10 nanograms per microgram.
Okay, yes. So, do you think that these cardiomyocytes, because these are like neonatal, they are like somehow productive, more productive than adults? Because 10 nanogram per ml TGF-beta is very high to induce fibrosis. Like in adult settings, we see that 10 nanogram per ml can induce fibrosis genes really quickly.
Sorry,
So the 10 nanogram per ml TGF-beta that you used is a very high concentration that can induce fibrosis genes in adult cardiomyocytes or in lung fibroblasts. But here you did not see any change in the fibroblast genes at 10 nanogram. Do you think that these cardiomyocytes, because neonatal, are protected somehow because these are like neonatal young population?
I don't know that, but I think the qPCR, I think primarily could have been user error. That's why it caused that. Because I think we have seen an increase in other people who've done this, use this control. So, yeah, I think it was primarily user error, but I'm still learning.
Learning very well.